Let's talk about fermentation. So we've already talked about aerobic cell respiration, and that's going to dominate in places that have oxygen available just because of the delta G that's available to the bacteria when they use oxygen to strip electrons off of various electron donors that are available. But when oxygen's gone, then if a microbe's going to survive, it either needs to respire anaerobically, and we talked about that in the last video, where an alternative terminal electron acceptor, something in place of oxygen, substitutes for oxygen, gives us less of a delta E and therefore less of a delta G and therefore ultimately less ATP, or they can ferment. Now, we mentioned in the last video that only prokaryotes can respire anaerobically, but there are examples in virtually every nook of life of fermenters. There are microbial fermenters that are macro fermenters. You and I can ferment, though not very well and not for very long. Our muscles can switch over to a homolactic fermentation when oxygen is not available. But there are a lot of definitions in use about fermentation, and so for a microbiology course, we need to make sure we've got a good, clear one. So here are 5 that are in circulation that I come across often, and I want to kind of talk about these and make sure that we understand that number 5 is the best definition for us. So frequently, especially in the food industry, you'll hear the term fermentation used to refer to spoilage of food by a microorganism. Whether the actual biochemistry of fermentation took place doesn't really matter. Someone in the food industry might say, okay, the mayonnaise has fermented, the potato salad has fermented, you name your favorite food that gets spoiled by microbes, and it's sometimes referred to as a fermentation. And of course, we don't actually know what biochemistry is taking place there. Fermentation is also used to talk about producing alcoholic beverages and acidic dairy products. In many cases, those are biochemical fermentations, but in some cases they're not, and it actually doesn't matter whether they are or not. Again, the food industry uses that term fermentation to refer to producing alcoholic beverages or acidic dairy products or kombucha and other weird things like that. In industry, there's a shocking number of products that either we as consumers use or other industries use that are produced by microorganisms. When those get scaled up to large vats, they're referred to as fermentations, and the vat is referred to as a fermenter. Again, regardless of the biochemistry that's taking place. Now, number 4 is probably the most insidious one, and this one unfortunately leaks into lots of textbooks. The implication is that any anaerobic energy-yielding process is called a fermentation, and so that would include anaerobic respiration. But you've just seen anaerobic respiration in the last video, and you know better. You know that that requires an electron transport chain of some kind, and it requires exogenous electron acceptors. Whereas fermentation, from a pure chemistry standpoint, is just a disproportionation reaction, where there's only a single molecule that's acting as both electron donor and electron acceptor, where the electrons are pulled off of one portion of the molecule and stuck back into another portion of the original atoms of that original molecule. So, number 5, we're going to use that as our fermentation definition. Any anaerobic or an anaerobic energy-yielding process doesn't use an electron transport system. That's your easiest way to remember. It's a redox reaction that's self-contained within the molecule, right? Glucose, for example, being fermented, acts as both the donor and the acceptor. So let's see if we can take a look at how that would look biochemically. Now, I had shown you a figure like this in class, a summary of glycolysis, the pyruvate dehydrogenase linking reaction, the Krebs cycle, and the electron transport chain. Let's say we're fermenting glucose. If we're fermenting glucose and there's no oxygen available and anaerobic respiration is not an option, then not only does the electron transport chain shut down, but also the Krebs cycle shuts down and our pyruvate dehydrogenase step shuts down. So now we've got glucose being split into 2 pyruvates. We get 2 NADHs and 2 ATPs. Not exactly a winning system here, right? If we could respire this with oxygen, we'd be mid to high 30s of ATPs. Without oxygen, some alternative electron acceptor, we might be a little bit lower than that, but certainly we're going to be able to do better than just 2 ATPs per glucose. So fermentation is not a highly energy-yielding process, but because it's only glycolysis, it's very rapid. And so bacteria that are fermenters have a competitive advantage in many cases over the anaerobic respirers just due to their turnover rate. But if you look at this reaction on the left, think about what's going to limit this, right? We're producing 2 ATPs. Those ATPs can then be used by the cell, And as the energy in the ATP is used, we dephosphorylate it, create inorganic phosphate and ADP, and those become part of the pool again. They're available for glycolysis and other processes to rephosphorylate, right? What about our NADH? Put high energy electrons into NAD+, we convert it to NADH. We used to recycle our NAD+ at the electron transport chain, but now we don't have one. So where are those electrons going to go? Pretty soon the cell's going to use up all its NAD+, have no place to dump those electrons, and no way to recycle its NAD+, and all of fermentation is going to come to a stop. So how do we solve this? NAD+ being regenerated is really at the heart of fermentation. What we're going to do, I say we as if it's you and me, but what the bacteria are going to do is they're going to dump electrons from NADH back into, in this case, pyruvate. The original molecule, the original bonds, they're going to dump it right in there. Or they'll modify pyruvate, some derivative, and then dump the electrons in there. That recycles the NAD+, glycolysis can continue, and the cell can continue to eke out a living by fermentation to ATPs per glucose. Now, when we dump those electrons into pyruvate, depending on the species, we can get organic acids like lactic acid or acetic acid. We can get alcohols like ethanol and methanol, or we can get gases like hydrogen and methane, or in some cases, some species will produce combinations of these. Maybe some acids and alcohols, maybe a mixture of acids, maybe gases plus acids or gases plus alcohols, and so on. And then if we start fermenting other molecules besides glucose, it's the same principle where we split it in a single step, get some ATP by substrate level of phosphorylation, and then dump the electrons from our electron or reduced electron carrier like NADH back into the products of the initial splitting reaction. And the sky's the limit for the kinds of organic acids, alcohols, and gases that can be produced. So fermentation can be very diverse, but hopefully you understand from what we just talked about how it comes about and how the chemistry happens. So we're going to stop this video here, and I hope this was helpful. Feel free to play it back several times. Let me know if you have questions.